So here's your information biology test, Monday, April 23rd. Chapters 12 with section 1, 2, and 3, and chapter 13 with sections basically 1 and 2. A couple of questions from section 3, but certainly not a lot of information. Take this time now to work backwards. We left off with chapter 13 in section 2 on the genetic code. How do cells interpret the genetic code?
Do we write these down?
Sure. So this is what you learned last time we met. The genetic code is read one codon genetic code is read one codon or 3 bases. So one codon is 3 bases. 3. I'm going to make this a 3. 3 bases at a time. Each codon matches an anticodon on a tRNA which provides an amino acid. And that's what your picture is showing on page 371. Also, what is a codon? And what is an anticodon? The codon is a 3-base sequence. I like this word sequence on mRNA. Okay, so red I should make adenine. If red is adenine, what would the other? Green. So this green would be what? What is it? If this is adenine, this is adenine, and this is adenine. So what would the green ones be? Remember, we're making RNA. Okay, so I'm going to put a U for uracil. U, and U. And if I do that, then what are the blue ones? Okay, the blue ones are cytosine. If the blue ones are cytosine, what are the purple ones? Guanine. You get it? So now we have to come to label a codon. What is a codon? So the question is, where are the 3 base sequences on mRNA. What would be the first one? At the end here we have a stop codon, and that's going to be U, G, A. Okay, so there's our 1, 2, 3, 4, 5. Our sixth codon is a stop codon, and that's UGA. So how many of, we have 6 codons here, and they are 3 base sequences on a messenger RNA. One will be a 3 base sequence which matches or complements the sequence of mRNA bases. So I'm going to have one of you come up here, figure out what's going to be the complementary base, and write it in on the board here with the appropriate color as well. Yeah, let's see. Let me mark this so that you know what it is. This is the mRNA. Just put the color and the letter. So change the color, and right here, one here, one here, one here, for the corresponding bases. So if this is an A, this would be a what? Would it be a T?
C.
U.
It's going to be a U. So yeah, see, because we're talking about RNA now. DNA is the only thing with a thymine. So if it's an A, we have to put everybody.
U.
Everybody.
U.
Okay, so go ahead and do that, but make sure it's what color? Get the green. Good. Get the nice. Now draw U. Is there going to be another one on this anticodon? Very good. And then one more. If that's a C, the corresponding one will be excellent. G for Gabriela. Good. Give her a hand. She did a good job. Chris, so he put an AAC for the second anticodon on that tRNA. Hey, Susan, he put a GAG for the anticodon. This is the anticodon. Shenda, she put these 3 bases on the tRNA, and that is an anticodon. So, and then we have one more. Who's going to put? He put the last anticodon, which is GAA, and so there we have it. Now we need the proteins for these tRNA. And put the first one there as cysteine, and we'll have Gabby put the next one. Okay, there we have it. Glutamic acid, tyrosine, glutamic acid again, glutamine, and cysteine. And these are the amino acids that go with each tRNA or transfer RNA. It's called transfer RNA because they transfer the amino acids to this site for alignment to make the protein. Codon is a 3-base sequence on mRNA, and the anticodon is a 3-base sequence on tRNA. 1C asks you on page 371, using the table in figure 13.6, identify the amino acids specified by the codons. They give you 3 codons there, UGG, AAG, and UGC.